So my name is Ed Kelly, and I'm associate professor in the School of Pharmacy and Department of Pharmaceutics, and a member of the Kidney Research Institute. My lab is involved in studying how our organs respond to drugs and xenobiotics and environmental toxins. So I'm a toxicologist by training, and one of the main projects in our lab is understanding kidney organ function, and to actually look at this in response to drugs, and also importantly, how we can actually use alternative technologies to animals to understand human organ function. And towards this end, what we've actually been doing for the past 8 to 9 years with funding from the National Center for Advancing Translational Sciences is to make what we call human organs on chips, these so-called mimetics or avatars of human organs, these 3D microfluidic systems. And we use these to model human organ function as a surrogate. Typically what you do when you're doing studies in the lab is you might do a very simple system, what we call an in vitro system, in a 2D cell culture system like in a petri dish. And then the next step for that would then be doing studies in preclinical species to understand how do things respond in animals. And then from a drug development standpoint, the next step you do is to do clinical trials. And in the clinical trials, you're relying on those animal studies as well as the in vitro studies to understand what happens in people. And what we found is that those are not always predictive of what happens in humans. And so using these human organ chip systems, we hope to actually bridge that gap in terms of functionality and understanding of the complex biology. So what we're doing on kidney cells too in this launch is we're actually putting up these human kidney chips to understand kidney stone formation. It's the complex medical term for that is nephrolithiasis. And so what we're actually doing with our kidney chips is we're exposing them to what we call microcrystals that are composed of ions of calcium and oxalate. And those are the main components of kidney stones. Kidney stones themselves can be, several millimeters up to the size of a golf ball. And what we're actually doing there is that we're trying to understand what happens in a kidney chip system that are exposed to not kidney stones per se, but actually calcium oxalate microcrystals. These calcium oxalate microcrystals are the same component of what you see in a kidney stone, but they're much smaller in scale. That's a great question. So the reason why we're actually performing this experiment in the ISS in microgravity is that we are actually making these microcrystals in the lab. They're 10 to 50 microns in diameter, so very, very tiny grains of sand, not even visible to the naked eye. But even with that small size, they will actually still settle out in 1G in the lab. And we're actually performing these experiments in a 3D microfluidic system, which means essentially we're constantly flowing through liquids through the kidney chip. You try to perform this experiment in a 1G lab, those microcrystals will actually settle out and will not flow through the system. In microgravity, these microcrystals will stay in suspension just like they do in our kidneys. So we were initially funded by NCATS back in 2012 to make a kidney chip system. First, to use it as a model of understanding drug toxicity. We then were funded by the same agency to understand how the kidney responds to disease models, or kidney disease models. And congruent with that, in parallel with that, we actually were funded by NCATS and CASIS, the Center for Advancing Science in Space, to actually do disease modeling in the unique environment of microgravity. And so we've been doing this now for several years. We launched our first mission back in 2019, and this is our second mission which we are launching. So many years in the making. Science is very incremental. We learn about what seem like great leaps, but all those great leaps are built on many small steps. That's a great question. So about 10% of the general population will experience kidney stones, which are very, very painful. And we really have no way of predicting who is going to get a kidney stone until they present with a great deal of pain. And so one of the great advantages of using a kidney chip system is we can monitor not only what is actually happening in the kidney itself, but also what comes out of our kidney chip, which is essentially the equivalent of urine. And so to understand what's actually happening in the kidney itself with these microcrystals, to look at the so-called cellular level, to access a person's kidney, you'd actually have to do what's called a punch biopsy, which involves sticking a needle into your kidney. And that's not something that people are going to volunteer for. But people will volunteer to pee in a cup. And so what we can actually do with a kidney chip system is we can look at, in those very early stages of microcrystal exposure, what is happening at the cellular level in the kidney cells in the chip, but also what comes out of the kidney chip being a equivalent of urine. We can look what's actually happening in the urine of those chips. And because we don't really know what to look for in the urine of patients, by looking at the cells in the kidney chip, we can identify potential, what we call biomarkers or predictors of what's happening at the early stages of kidney stone formation before you're presenting with that excruciating pain. Absolutely, because what happens in microgravity is you have significant bone loss, which is what we were studying the role of the kidney in bone loss in our first mission. And when you have bone loss, what you're actually losing is you're losing the ions in your bone, which include calcium, which is the main positive ion, along with oxalate, which is the formation of kidney stones. And so in microgravity, you're having a lot more calcium going through your kidneys. And so because of that, astronauts will actually have an increased risk of kidney stones as well. And so by understanding what happens in microgravity and be able to identify potential biomarkers or predictors of kidney stone in microgravity for astronauts, we can actually put in interventions, whether that be increasing hydration, modifying their diet, or things like that. And that's very important because when it comes to people on Earth that get a kidney stone, maybe they have to take a week or two off of work and to get pain medication, etc., and they have to deal with that. But in the case of an astronaut, say on a mission to Mars, where you've got 4 or 5 astronauts, and you suddenly have one who's got kidney stones, and if it's severe enough and you need surgery, there's no option for surgery, and you're also down 20 to 25% mission efficiency. I mean, what's interesting to me about this is that we can understand what's going on at the cellular level in our kidneys, in microgravity, not only because that is very important for understanding kidney health terrestrially, but also what's happening in astronauts. Because I would like to think in my lifetime that we're going to see people back on the moon, men and women, and also a mission to Mars. And being able to understand how human organs respond to that, which is an important aspect of the human research program, is something that I'd like to think that chips in space, human kidney chips as well, will contribute to. I was 5 years old when I watched Apollo 11. Okay, I am a big sci-fi nerd. I'm a big Star Trek fan. I have a Borg cube in my office. I have the poster for Star Trek 6 in my lab, which is the undiscovered country, which to me is a metaphor for science. So yes, I have, beyond just the important scientific interest in this, I have a very strong nerd aspect to this as well. It is absolutely exciting and phenomenal. I never thought I'd ever get to do anything like this, so it's very, very exciting. But with the excitement, of course, comes the stress, because there are so many things that can go wrong that I don't even want to talk about that to jinx it. But so many things have to go right for this to go off. And when we launched back in 2019, we had many, many delays, which included, right before we were supposed to go launch, a failure of what I think was called the MBSU. And so with those sorts of stressors involved, I just want to think that I'm here at this time tomorrow and we see a beautiful launch. Other than the fact that kidney stones affect a large number of people, 10%, and if you get a kidney stone once, you're much more likely to get it again. So certainly immediate impacts are ability to identify, particularly in people at risk, early biomarkers, urinary biomarkers, to actually improve their health and prevent them having to get a second or third kidney stone.